I'm a little bit ahead of time, and so I'm going to take just 5 minutes to try and get a head start on cystic kidney disease because this is certainly a longer topic than magnesium, and so let's do that. Let's get a head start. Okay. Certainly, as you're talking about cystic kidney disease, the one that you absolutely need to know backwards and forwards is autosomal dominant polycystic kidney disease. You will take care of many patients over your career with ADPKD. This is deemed to be one of the more common monogenetic diseases, period, not monogenetic kidney diseases. I'm saying this is one of the more common monogenic monogenic diseases. So it is something that is relatively common. Now, the mutations are generally most commonly in a gene called PKD1, which codes for polycystin. Now, you can have a mutation in the PKD2 gene in a lesser proportion of patients. And then keep in mind that there are other less common mutations in other genes that can also cause ADPKD. So even though you may consider most patients as PKD1 or PKD2, in fact, there are other genes that can also cause an autosomal dominant cystic kidney disease. In general, those mutations cause a milder phenotype. In general, mutations in PKD2 also cause a milder phenotype. Okay. Now, there is some genotype phenotype correlation, not only between whether you have PKD1 or IFT 140 mutations or PKD2 mutations, but the amount of gene that's abnormal. So if you have a truncating mutation, that creates a very small yeah, gene with a small transcript that essentially is totally non-functional, those patients would tend to have more severe disease manifestations as well. These genes in general code for the cilia or primary cilia that is present in these tubular epithelial cells lining the entire length of the nephron. Okay, essentially all epithelial cells have these primary cilia. And you can have dysregulation in cell division throughout the entire nephron unit, okay, from Bowman's capsule to the deep medulla. Okay, so those are all places where you can have, again, uncontrolled cell division resulting in the cystic dilatation of the nephron, and then ultimately macrocysts that can be seen on imaging. Okay, um, so again, there is this genotype phenotype correlation. There will be the rare patient who has yet another more severe manifestation where they may have a mutation that extends from the PKD gene to a neighboring gene, the TSC1 gene, which mediates tuberous sclerosis. So these individuals will have a very aggressive phenotype in general, resulting in ESRD even as a teenager, and they will have a blend of both diseases. We'll go over the phenotype of tuberous sclerosis in next week. Okay, just a little bit more about PKD1. Um, Again, this mutation in ciliary biology or mutation that results in altered ciliary biology seems to cause a dysregulation of epithelial transport, creating, when I cannot sense the luminal flow, that epithelium will then either alter its reabsorption or secretion of tubular solute and fluid, resulting ultimately in some of these macrocysts. They would also have altered cell division as well, resulting in, again, the creation of these cystic dilatations of the kidney throughout the entire length of the nephron. Now, what will become relevant when we talk about therapeutics are that this tubular secretion or transport, let's use a more general term, is mediated um, through the CAMP signaling. Okay, so this is GTPases that are that have cyclic AMP signaling downstream, and so we are leveraging this by blocking vasopressin signaling through tolvaptan. That seems to influence the downstream signaling through cyclic AMP and therefore modify disease expression. So reduce cyst progression we think of by downregulating cyclic AMP mediated signaling. Okay. There's also some biology that centers about the chloride channel that is coded by the CFTR gene, the same gene that mediates uh, um, cystic fibrosis. And so there's some basic science that looked at trying to inhibit some of the um, altered transport processes that arise from these CFTR gene products, in order to modify PKD disease progression that really has not hit human trials, but was experimented or was relevant in animal models of disease.